Welcome to this edition of CellTalk presentation. Our presenter today is Jay Darji. Uh, just to give a brief introduction of his, Jay is an engineer with well-developed skills in data analytics, research, and software development. He is also a good communicator with good interpersonal skills and is used to working in a team. He is always enthusiastic to learn and undertake new challenges. At Renix Life Sciences, he works as a junior engineer working in a technical research department for digital medicine. He is also responsible for developing various statistical methods for anomaly detection with human heart rate data captured with the help of wearable devices. He is an electrical engineer. He did his bachelor's degree in electrical engineering from the Institute of Infrastructure Technology Research and Management, along with voluntary work at the Blind People's Association. Moreover, he has done research in biomedical signal processing and machine learning as well. Since the completion of his studies, he has wanted to become a data scientist. Thus, he began learning data analytics and machine learning through a summer internship at Renix. He hopes to increase his technical knowledge while providing solutions and improvements to common industry problems. In the long run, he aspires to contribute significantly to industry development and optimization to create something that would genuinely improve the lives of thousands. Uh, I asked Jay Darji to start his presentation.
Thank you, Dr. Vijay, for the introduction. Hello, everyone. I am Jay Darji, and I'll be presenting about the methylation aging clock based on deep learning. So, the original author for selected paper is Fedor Gelkin et al. The paper title is DeepMAge, a methylation aging clock developed with deep learning. The topic selected was longevity, and this paper was published in a journal called Aging and Diseases in the year 2021, and the journal having an impact factor of 7.4. This research has an impact on biological aging of organs, longevity and quality of life, and it has an impact of anti-aging therapies. So let's start with the introduction. So we all know that longevity refers to the actual duration of individual's life with focus on understanding and extending the human lifespan. Aging can be influenced by various factors such as environmental, biochemical, evolutionary constraints. In biogerontology, deep learning methods have been used to create number of novel age predictors, which can be used to develop geroprotective interventions or to help aging conscious people understand and potentially affect their pace of aging. Aging clocks have proven to be invaluable tool for biogerontology since they often offer a unique opportunity to quantify the aging process. So there is a general background of it that DNA methylation aging clocks have first introduced in 2013, where there were two studies. Both describes two different algorithm which is predicting the chronological aging of a human. And since then, it has become an invaluable tool in biogerontology. So after that, after 2013, the variety of machine learning approaches have been tested to predict human aging based on molecular level features. And among all these, deep learning or neural networks are the promising approach that has been used to construct accurate clocks using blood biochemistry, transcriptomics data, and other microbiomics data. So the present challenge is in today's world, aging clocks can predict biological age, but to identify which specific molecular or physiological factor contributes to the aging process is challenging. And understanding the causative factors can lead to more targeted interventions and therapies. So the objective of this research is to explore the performance of deep learning in DNA methylation settings and compare it to the current industry standards. This is the methodology for current study. In this study, the data have been collected from Expression Omnibus repository. It consists of 32 studies with 6,411 DNA methylation profiles. And among these, 17 studies with 4,930 samples were in training set, and the rest 1,293 profiles were used in verification set. All the data which have been used in this study is collected using Infinium Human Methylation 450K and 27K batch chip platform. And only studies with available age metadata and raw files were selected, and the rest were excluded. In the pre-processing step, the LumiR package was used for intra-study color correlation and normalization. And only 24,538 CPG sites shared between the 450K and 27K platforms were used. And the sex chromosome sites and the sites with orthologous sequences on multiple chromosomes were excluded from the study. In all the profiles, around 17% uh, samples associated with the project reported the integer age values, and to avoid the bias into the model, the 0.5 years count were added to the integer ages. So after collecting the data, they replicated the 2013 research, which is Howard clock, and they used the same 353 regression coefficient from that research to create a linear regression model. And after that, it was used to estimate the logarithmically transformed age. And additionally, a novel elastic net model was trained using protocols from the same research. Elastic net is uh, it is used to restrict the coefficients to take the extreme values. It is basically a kind of penalizing term. After that, they constructed a deep neural network. They uh, they created a deep neural network to allow fitting the data with high dependencies, and this takes this takes input as a DNA methylation beta vectors and gives continuous age as the output. The model was trained on 24,538 features, the same CPG features, and because of the high dimensionalities, the feature selection method was applied. After that, the gradient-based feature selection was applied, and this was applied to find the important features out of all the 24,000 features. So for model, optimal parameters were obtained using a grid search algorithm. And at the end, the final model was trained on 1,000 most important features with five-fold cross-validation. And the loss function mean absolute error was minimized using backpropagation algorithm. The final neural network consisted of 4 hidden layers, and each layer had 512 neurons. So in the results section, this particular study, DeepMAge, used a collection of around 5,000 blood DNA methylation profiles and obtained a cross-validation median absolute error of 2.24 years from the controlled cohort of 17 studies. Control cohorts is nothing but the healthy subjects or healthy healthy person. And in the testing set, there were around 15 data sets with having 1,293 samples, which was slightly less accurate than the training set, with having a median absolute error of 2.77 years. In the case cohorts, which means the patients with having some illness, the median absolute error was 3.29 years in training, and whereas it was around 4.18 years in a verification samples. So after that, this study was uh, tested on 15 different data sets which were having illnesses. So after that, authors have identified that the study on tauopathic frontotemporal dementia and palsy cases were the cases were 1 year older than the control cohort. People with inflammatory bowel disease were predicted by DeepMAge to be 1.23 years older than the controls, and women with ovarian cancer were predicted to be 1.7 years older. Then multiple sclerosis patients were predicted to be 2.10 years older. People with congenital charge and Kabuki syndromes were quite interestingly predicted to be 5.2 years younger than the normal subjects. Then congenital hypo hypopituitarism was associated with predictions of 5.6 years older than the prediction of control cohort. And this result indicates that the faster pace of aging in people with these pathologies, except the charge and Kabuki syndromes. So after that, the results of DeepMAge was compared to the gold standard 353 CPG aging clock. So authors found that the deep learning based methylation aging clock was accurate and outperformed the 353 CPG clock and the 71 CPG clock. So the original 353 CPG clock had the median absolute error of 3.56 years, and the same model which was reproduced by the authors having the median absolute error of 3.151 years. The correlation between predictions by 353 CPG clock and deep learning based methylation aging clock for verification set was slightly high, and the Pearson correlation for these two clocks is 0.96. So authors found that the deep learning based methylation aging clock is more accurate in predicting the age of healthy individuals than 353 CPG clock, and also with 71 CPG DNM methylation aging clock. The 100 features that constitute the DeepMAge contain 121 CPG sites from the original 353 CPG clock and 75 from the one they reproduced. The genes were the DeepMAge CPGs were located are enriched with those taking part in developmental process, especially cardio and neurodevelopmental processes. So authors hypothesized that this may be explained by antagonistic pleiotropy theory of aging. According to this theory, genes required for the early staging of development may sustain their activity beyond their appropriate period of expression. This non-specific activity harms the organism and leads to multiple downstream after effects of ultimately manifesting as aging. So this is the first deep learning DNA methylation aging clock that outperforms the linear models in the literature. And although the biological relevance of deep image has been determined in multiple case controlled independent data sets, its mortality association is yet to be established. So in the conclusion, epigenetic aging has numerous quantitative models. They developed uh, The authors developed DeepMAge that is able to predict the age with precision. The neural networks can be used to explore individual DNA methylation landscapes in the context of aging, and they can potentially be used to estimate the risk of certain aging related events in the future given a single observation. The impact of this research is that this deep learning based methylation aging clock is sensitive to diseases, a property that makes it valuable and as a potential health marker. And DNA methylation clocks can be used as biomarkers for healthy and unhealthy aging, as well as be used to predict diseases at risk. So thank you everyone for patient listening. And if you have any doubts, you can find me at jay@renix.com.org.
Thank you, Jay, for this illuminating talk. Questions are welcome. Nupur Viswas has a question. Do they explore individuals with any comorbidity?
No, I don't think.
Okay, thank you, Jay. I hope that answers Nupur's question. I don't see any further questions. Can you please go to the last slide?
Sure.
Our next presenter will be Yogita Das, and she will be presenting on comparison of long-term effects of metformin on longevity between people with type 2 diabetes and matched non-diabetic controls. Our presentation will be on 6 December 2023. I thank everyone who has attended today's CellTalk. Thank you, everyone.